Ladies and gentlemen, it gives me great pleasure to welcome you to our next panel focusing on therapeutic strategies to prevent and treat COVID-19. I'm Max Herrmann, Head of European Pharmaceutical and Biotech Research for the US investment bank Stifel, and based here in London. Co-chairing today's panel gives me great pleasure to welcome Movva, Novartis' distinguished scientist and an independent pharma and biotech advisor. We also have four other learned members of our panel today. Dave Hallett, Chief Operating Officer of Exscientia Limited. Eddie Littler, founder of the Collaborative Community Against Coronavirus, who is also Chief Operating Officer of ReViral. Gilead Raday, Chief Operating Officer of RedHill Biopharma. And Richard Marsden, Chief Executive Officer of Synairgen. On today's panel, we'll be discussing the repurposing and re-engineering of existing drugs, exploring the roles of different pharmaceutical modalities, be they monoclonal antibodies, antivirals, and immunomodulators, developing drugs specifically, so new drugs against COVID-19, and the interplay of therapeutics with potential vaccines, and then thoughts on prophylaxis versus early treatment versus late treatment. We've seen Gilead's remdesivir offer benefits in the speed of recovery for hospitalized patients from 15 to 11 days, a 31% improvement, although the 1,000 patient or thereabout study narrowly missed showing a clear mortality benefit. We've seen the old steroid dexamethasone demonstrate an almost 30% reduction in mortality in patients with COVID-19 on ventilation, and 11% for those patients on oxygen support. And later we'll hear from Richard Marsden, CEO of Synairgen, whose inhaled beta-interferon-based therapy, SGN-001, reduced the risk of death or progression to ventilation in hospitalized COVID patients by 79%, a significant result, even though the study was only 100 or so patients. And most recently, we've seen new data from Lilly's JAK kinase inhibitor, Olumiant, show benefits in hospital recovery times when added to remdesivir. So there's an enormous amount going on in the therapeutics field. So therefore, it gives me great pleasure this morning to introduce our first presenter, Eddie Littler, from the collaboration of Community Against Coronavirus. Thanks. Over to you, Eddie.
Thanks, Max, and good afternoon or good morning to everyone on the call. I'm going to do this quite quickly. We have ample opportunity later on to answer questions. So this sets the scene. In the world over the last 15 years, we've seen 5 viruses emerging. These include Ebola and Zika, but the other 3 are coronaviruses, which we're all familiar with. In fact, it's worthwhile taking a few minutes to think of the fact that there are over 500 coronaviruses identified in bats, and many others identified in other animal species. What's unusual about coronaviruses is that they have the ability to repair their genome, and this gives them the potential to almost recombine. And I think it's worthwhile saying we've all seen the evidence now that the coronavirus family is pandemic ready, and there probably will be more pandemics with coronaviruses in the future. I've summarized here the approaches that Max has just introduced us all to. And I think for SARS-CoV-2, there are frontline therapies that described above, but the time course we have will limit which of these new agents are going to be developed. Have the next slide, please. So our approach in 3C were founded by a group of experienced antiviral drug hunters from big pharma background and also biotech. And you can see our pedigrees here. There's 2 virologists and 2 medicinal chemists. We're what's called in UK a not-for-profit company. We've been funded so far by individuals and 1 or 2 university organizations. And our strategy is to target the coronavirus family polymerase, which is highly conserved. And we think by targeting this particular enzyme, we can end up with inhibitors that cross the family. I think it's also important when thinking about this for the next generation compounds to focus on compounds that can be made cheaply, so they can be used in developing countries and also stockpiled. We started with a few compound collections that were designed by 1 or 2 of us in the 3C company, specifically for viral polymerases, and have been screened before in some of our parental organizations shown above to screen for virus inhibitors such as hepatitis C. We ended up with around about 10,000 compounds. Have the next slide, please. So where we are today, I said before about the 3C status. We've screened about 8,000 of the 10,000 compounds. We've got a large number of hits. To a certain extent, the number of hits were a little bit surprisingly large, but since we have a very specific library where we would anticipate seeing hits, maybe it's not so much a surprise. They've good potency and good toxicity. They're very good starting points for drug discovery programs. And we've started generating new chemical entities based around these hit molecules. Our plan is to seek further investment and take these hits right the way through to IND. Then we will out-license to pharma or NGOs for clinical development, and any profits that we do generate will be reinvested into company to find inhibitors of other viruses. So we're now out seeking further investment to cover the lead optimization of the hits that we found, and these are from other high net worth individuals or other sponsors. And as it says in the last plea, we will of course accept checks or even PayPal. Max, over to you.
Thanks, Eddie. Right, now from Eddie's non-for-profit group, we'll head over to Gilead Raday, Chief Operating Officer of RedHill Biopharma. Over to you, Gilead.
Thank you, Max, and pleasure to be here today on the panel. Next slide, please. RedHill is publicly listed, and the presentation contains forward-looking statements, so the standard representation or warranty is made as to the accuracy and completeness of this presentation. So a little bit of background. RedHill Biopharma is an emerging US specialty pharma focused on GI and anti-infective drug development and commercialization. We're currently promoting in the US 3 commercial products, and we have a broad and advanced pipeline, phase 3 programs in multiple indications. And down at the bottom, you can see we have 2 programs that have been positioned and realigned to development for COVID-19. And they're really some of the front runners now in terms of the stage of their development. And I'll go into a few more details. Next slide, please. So again, 2 shots on goal within the company from our internal new molecules. Our front runner lead program is opaganib, which is a novel, unique, dual-acting drug which has both antiviral activity and anti-inflammatory activity. And we'll go into more details on that. It is currently ongoing global phase 2/3 study. And of course, the name of the game is time. And we're working very rapidly towards enrolling that study with the hope of getting all the patients enrolled by the end of this year. Our second shot and product, second runner, is upanostat, our HB107, which is a serine protease inhibitor, also with antiviral activity and additional supportive activity, which can be to support lung damage. Both of these compounds, importantly, are orally administered, which are give them the attribute of potentially spanning the use from mild cases to severe cases. And I'll go into more details on the next slide for opaganib. So opaganib really is a very promising product from the information we have to date. The mechanism of action combines very potent antiviral activity against SARS-CoV-2 and anti-inflammatory activities on top of that, which we knew from prior development. So what we see here is that in a recent study of in vitro model of human bronchial tissue, opaganib completely inhibited SARS-CoV-2 replication, and compared favorably in this study as compared with remdesivir, which was the active control of the study. So again, this is very encouraging, and we can see that from 1 microgram per milliliter, the effect is maintained, and this is pharmacologically relevant concentration for the doses that we can provide to patients with the oral dosing of opaganib, which is known to be safe. Next slide, please. I would also mention just that because it's targeting the sphingosine kinase 2, which is a cellular human host factor, we have a better likelihood of avoiding resistance developing than therapies that target the actual virus itself, which can mutate more rapidly. So on top of this antiviral activity, which is very potent, we already have evidence from use in humans in compassionate use cases where there was a real benefit to patients as compared to matched case controls from the same hospital, with none of the patients that were given opaganib progressing to ventilation, while a third of the control group progressed to ventilation. The median time was reduced from weaning from oxygen to 10 days as compared to 15 days, and additional supportive markers, inflammatory and other, confirm the activity in these patients. So we're now again focused on getting to the potential emergency use authorization, hopefully before the end of this year, by completing the rigorous phase 2-3 studies that are ongoing in multiple countries globally. The studies have been approved in the UK, Russia, Israel, Italy, and Mexico, and we're expanding because really we feel that it's important to address the needs of the pandemic as quickly as possible. So we're also preparing for ramp up of manufacturing. As you said, being able to distribute and scale up and build up inventory is critical to be able to support global demand. And we're working with US government agencies in order to potentially support us in getting there. Next slide. Thank you, and back to you, Max.
Great, thank you, Gilead. Now it gives me great pleasure to introduce David Hallett, Chief Operating Officer of Exscientia. Over to you, David.
Thank you, Max. Perhaps we get straight into it. The next slide, please. So very briefly, Exscientia is a privately funded organization. It's actually founded back in 2012 as a spin-out from the University of Dundee by our current chief exec and founder, Professor Andrew Hopkins. We are at the forefront of AI-driven small molecule drug discovery. And to the best of our knowledge, still the only company that's actually successfully designed molecules entirely by AI that are in clinical studies. And the little kind of picture on the left highlights a press release from our partners, Dynapon Sumitomo, back in January of this year. We're also due to take an A2A antagonist that was designed through a collaboration with Biotech into first-in-human studies in the fourth quarter of this year. We're currently 70 scientists based mostly in Oxford and Dundee, but we do have a physical presence in both Japan and the United States, and we continue to expand rapidly even during these challenging times. Next slide, please. So as I think Eddie outlined very eloquently, there are two approaches that we've taken at Exscientia. One is summarized in this slide, which is that what can we do to help now? So in terms of short-term impact, i.e. repurposing. So at our laboratories in Oxford, where I'm actually kind of based today, is that thanks to assistance from the Gates Foundation and from Calibre, we actually got hold of the reframe library from Scripps, and actually screen that against two of the key proteases within COVID-19, namely the main protease and the papan-like protease. Just to orientate you, the reframe libraries is a library of 12,000 or so therapeutic agents, many of which actually are on the market and which all have minimally undergone some form of preclinical safety tox studies, meaning that if you find a viable hit, is that there is a mechanism to take that rapidly into human testing. From this work, we identified a number of potent inhibitors against the main protease, with the best one having an IC50 in around about the 30 nanomolar range. And together with our kind of collaborators at the Diamond Light Source and both the Oxford Structural Biology and Oxford Departments of Chemistry, we're able to kind of generate kind of crystal structures for these molecules. And just as importantly, we've actually identified the first, to our knowledge, reported inhibitor of the papan-like protease with an IC50 of about 300 nanomolar. I can't tell you what those projects are today, but we just submitted a full paper to Nature, and we're actually having conversations with the license holders at the moment. Next slide, please. In terms of long-term preparedness, as Eddie pointed out, this because we've seen beta coronaviruses like this before through SARS and through MERS and through COVID-19. This is not a kind of a chance finding. It will happen again. So I just want to introduce you to the CARE Consortium, of which Exscientia is one of the members. So this is a large European research initiative funded through a variety of mechanisms as a kind of public-private partnership. It's actually worth over 74 million euros over the next 5 years. And it does combine a rapid emergency response as well as with a long-term preparedness, i.e. trying to find agents that would be available in the marketplace 5, 6, 7 years from now that would have pan-coronaviral activity. Next slide, please. And in terms of the emergency response, is that there is a drug repositioning arm, and the findings that I just alluded to at Accenture have been fed into this. But there is a de novo small molecule drug discovery effort, and Accenture is actually providing the AI-driven small molecule design to this consortium, and we're actually leading the hit to candidate work package with the University of Leuven. And the overall goal is to identify 2 to 3 effective, safe development candidates that have demonstrated broad spectrum coronaviral activity. So like Eddie, we're looking at kind of those viral targets which have been kind of heavily conserved over time, and therefore providing a likely opportunity to be able to kind of to treat potential coronavirus outbreaks in the future. And that's my last slide.
Right, thank you very much, David. And now it gives me great pleasure to introduce Richard Marsden, Chief Executive Officer of Synairgen, for our last presenting panel member. Over to you, Richard.
So as a company, we've been developing a broad spectrum antiviral, which we're delivering directly to the lungs via aerosol to treat severe viral lung infections. The drug is interferon beta. Interferon beta is well known as a drug that's given by injection to multiple sclerosis patients. But if you ask biologists what interferon beta does, they would hopefully tell you that it drives our innate immune response when we bump into viruses. So as a drug, it's got potential to be used every year to treat patients hospitalized due to respiratory virus infections, obviously severe enough that's put them in hospital. And there's a whole range of viruses that where this drug could be used. Influenza gets the headlines, RSV close behind, but the list is long and includes parainfluenza, human rhinovirus, adenovirus, regular coronavirus. So forgetting for now this huge annual business that's where this drug could be used and focusing straight onto coronavirus, it gets quite interesting. So the reason we are developing it for the annual business is that older patients and some patients with comorbidities don't make enough interferon beta when they bump into viruses. So that's reason enough to start to think about giving it to coronavirus infected patients. But this particular coronavirus has got a trick up its sleeve that it uses to evade the immune system. And that is that it suppresses interferon beta production. And there's quite a literature around this now. So that gives us 2 reasons to want to deliver this drug to patients with COVID-19. You can't use any old interferon beta formulation, though, and we have developed a pH neutral formulation, which is free of some of the excipients which could be active in the airways. And it's delivered by highly efficient mesh nebulizers. In our clinical trials to date, we've dosed nearly 300 patients who are asthmatic and COPD patients and now COVID-19 patients. And we can activate antiviral pathways in the lungs, which we demonstrate by looking at the gene activity in sputum cells 24 hours after dose delivery. That's the background. Next slide, please. 2 slides out of many that I could have shown you from a 100-patient pilot study that we conducted in April and May this year. This is in the UK, and it's in COVID-19 hospitalized patients who were either hospitalized and coping with room air or required oxygen. We looked at several endpoints as coached by WHO guidance issued in February this year. I picked out 2 here. We're quite excited by a acceleration or improvement in the chances of recovery. Recovery here is defined as getting back to normal or no limitation of activities, and you are more than twice as likely to recover on drug compared to placebo. We looked at various other endpoints that use the ordinal scale, and Max cited one of them earlier, But as a respiratory company, we get quite excited by breathlessness as an endpoint. And you can see a marked reduction in breathlessness in patients treated with interferon beta or SNG001, our special formulation of it. Next slide, please. So we've got a drug here that is well established, safe given by injection. So far in nearly 300 patients, very well tolerated, given by inhalation with this special formulation. It's got an annual market to treat people hospitalized due to a whole range of respiratory viruses, which range in patient groups from the very old to the very young to the immunocompromised, or there's just those who've just been unlucky and not coped with a respiratory virus very well and ended up in hospital. Right here, right now, we're very focused on progressing this drug as quickly as possible towards a treatment for patients hospitalized due to COVID-19. We're also looking at home-based population to try and keep people out of hospital. And we're super focused on this recovery, or getting people back to normal as quickly as possible. Apart from clinical trial activity, we're also investing in the manufacturing so that we have hundreds of thousands of treatments being manufactured per month in the new year.
Okay, Richard, thank you very much. I think that was an exciting set of presentations, and I hear a lot of good news coming from various small molecules coming from some time where we have started with COVID. We have no clue where this virus is going and how the patients are suffering, and I see a lot of good news. So we heard about a couple of small molecules today, and we heard about a biological. There are many other efforts. Vaccines I don't go into, but there is also efforts at the antibody level that patients can be treated. So with all these things, there is a lot of hope. Nevertheless, I want to ask a question to Gilead as to, with all these things between antibodies and antivirals and the biologicals and small molecules, do you think we need all of them? Or do you think each one has a role to play in the subset of patients? What are your thoughts on this, Gilead?
Oh, thank you, Rao. I think there's certainly uncertainty as to what molecule is going to come out most effective or most helpful, but I think there's room for several approaches. Small molecules like we're developing have, as I mentioned, advantages of being orally available and being able to be easily administered to broad patient populations, from mild cases outside of settings where they're being taken care of, all the way through to if activity is potent enough, and we see that there is potent activity in some of the small molecules, all the way through to the severe cases that are hospitalized. So there's certainly room for other approaches, because many of the severe cases do get hospitalized and require intensive care. So certainly a lot of different approaches can eventually come out and support these patients.
Okay, thank you.
Right. Maybe I'll ask the next question, and for Eddie, we've heard obviously a lot about remdesivir and dexamethasone as two early repurposed and re-engineered drugs. I wondered what your thoughts were about what those two therapies and their effect on COVID tells us about the disease and the potential for future therapies.
Yeah, thanks, Max. I think what it tells us is fairly clear, that there's a role for both antivirals and immune modulators in treating this disease. SARS-CoV-2 obviously is a classical virus resembling RSV in some cases, where you get virus replication that induces an immune response. Sometimes people summarize it as cytokine storm. I was around in Wellcome when HIV broke out, and where we repurposed, it wasn't quite that way, AZT. And that came through very quickly. It was the first medicine we got into patients, and it did help patients. People forget that, for a period of time, but it was not the ideal medicine. So I think that's one thing we need to look at. We're probably in the beginning of an evolution where the first repurposed drugs will bring as much relief as we can to patients, both knocking down viral load and treating symptoms. But later on, hopefully, we'll have more dedicated drugs which have more potency and perhaps a better safety profile.
And then maybe, David, do you think that that there can be further drugs developed from the existing portfolio, and how much time do you think there will be in order to do that from these existing drugs?
So I do believe that that we can improve upon, and what's called this first generation of kind of repurposed drugs, as Eddie describes them. I think we're learning about kind of both their effectiveness, but also kind of where perhaps they've got some flaws, because there may be areas where they weren't optimized for this particular kind of scenario. I think in terms of the time frame, I think what we're learning is that as we come together as a community, is that we can accelerate both drug and vaccine development. So I am optimistic that the traditional timelines of kind of first in man to marketed drug of up to a decade in some cases will be accelerated in this case. But I do think it will still, in order to kind of come up with a future medicine that's targeting a broad coronavirus population, not just effective against COVID-19, I still think it will take kind of 4 or 5 years to identify a molecule, take it through a variety of kind of trial settings in different geographical locations against different kind of forms of coronavirus, to kind of hand on heart say that this is future preparedness. Here's a molecule that will have utility against a future pandemic.
Okay, thank you for that answer. And I have another question. Getting back to the COVID-19, as we all know, there are some molecules that have shown some benefits, but many new molecules have jumped into the scene and they are being developed. Vaccines are just around the corners, some people say, and though we haven't seen the first successes yet. So given the fact that if vaccine is successful, they are cheaper and accessible readily for most of the people, what do you think? This is a question to Eddie that I can ask. What do you think the therapeutics will play a role in the future? Will they be relevant, or will they be just useful for only a small subset of patients?
Well, the way I'd like to answer your question is to refer to what David said, is that our strategy is to look for a pan-coronavirus inhibitor. Frankly, if you haven't got a molecule in clinical development now, you're probably not going to have a major impact on SARS-CoV-2. Now, when you look at trying to develop a pan-coronavirus inhibitor, a vaccine is very limited. If you look at examples of flu, we have to develop a new vaccine each year. So I think that's where you basically look at a new inhibitor will be a molecule you can get into patients very quickly, dampen down the spread of the virus and the effects of the virus, and allowing society to prepare itself better for the next pandemic coming through. So I think that's going to be the big use of the inhibitors, is broad spectrum.
Okay, great.
And then how, this is maybe for Richard, how do you see the vaccine and the therapeutic world evolving for COVID-19 specifically? Do you see, if we hopefully are lucky enough to have a vaccine, that will be the end of therapeutic use, or will there be a window of opportunity still for therapeutics?
I think we have to think about how much we know about this virus and how much we don't know about this virus. It's so young, and anyone who says anything with any great certainty is probably going to be proved to be wrong. I think we have to hope that a vaccine will come along, or more than one vaccine will come along sometime soon. And if one looked like it was doing some good, I'd be jumping into the queue to get it. Even with good vaccines, you get people who don't respond particularly well to the vaccine. And, with influenza, every year it appears that a lot of older people don't get the full benefit that we'd like them to have from influenza vaccines. And that may turn out to be the case with coronavirus. We also don't know whether the virus will change itself, so the vaccines don't work so well. So I think and some people won't get vaccinated for whatever reason. And so I think there's always going to be people who need help beyond what a vaccine can do for them. And we have to be adaptable with all the therapeutic approaches we can have. And then, what happens if it isn't coronavirus that comes along next time? You need broad spectrum therapeutics that can help people. This time around, it's the lungs which are under attack, although you do get complications in other organs. You know, we've had Ebola not particularly affecting the lungs. So we, as humans, we have to be on standby and ready for these events. And you know, enough people have been talking about the bad one just being around the corner. Well, it's hit, and no one alive today you know, has got any previous experience that's helpful right now. So we're all in this together, fighting this one.
Yeah.
Okay, along the same lines, I would also like to ask the panel a very general question, but first I'll direct this to Eddie and Richard, and other people can chip in. So we have the virus today, and unfortunately we don't have a way of preventing it, but we are still treating the people in an acute way, in many different ways, but we still haven't addressed the issue of whether any of the subset of patients actually need chronic treatment lifelong, because the virus does substantial damage in a subset of patients. Okay, Eddie and Richard, have you thought about these things? Are there some of these treatments that we are putting into play? Some of them will help acutely, some of them might be administered lifelong.
I can comment on the way we've seen it. We got super excited when our data came through. It's a small trial, but we had this encouraging effect on changing the odds of progression to severe disease. But over the last 2 months, we've got more and more excited about this encouraging signal on getting people back to normal, getting people back to no limitation of activities. But who knows what's going on? Why does this virus have this effect on some people? And I know that a lot of academics around us are really gathering together to try and understand what's going on, to try and help handle the virus better when it is there. But even when the virus is gone, why do we get this long-term unwanted effects that are so debilitating for people?
Yeah,
it's an unknown.
Okay, are there any questions from the audience? Max, I don't see a blip on my screen.
No, I think if you want and you're in the audience, there is an opportunity to ask questions through the chat comment at the bottom. But in the interim, I guess I'd be interested from Eddie, you're a virologist and you've been developing antivirals many years. I'm interested in the activity that you've seen from remdesivir. Does that sort of imply that if you had a more potent molecule, you could get very significantly better results, or are you still going to have to get early, you know, very safe molecule that you treat people very early in the infection cycle?
Yeah, I think remdesivir is going to be a good drug as an initial hit on coronavirus. And after all, we don't have very much else, but it does have limitations. You've got to be a bit careful quoting potency because it's variable between different cells in the lab. So monkey cells, it's not particularly active. Human cells, it's a lot more active. It's about activating the prodrug, and that can be misleading. But even so, it's not the most active drug I've ever seen, and it does have a therapeutic window. So there is some measure both in the laboratory and I understand in the clinic of some emerging adverse events. My suspicion is that remdesivir will not have the right profile to be used very early on in infection, prophylaxis, for example. And we will need much better, cleaner drugs to be used in a prophylactic setting. And after all, as I said before, that's when ideally you want to treat with an antiviral. Just look at the old data we do to flu. Tamiflu and Relenza. Relenza came out of my labs in Glaxo. You have to treat within 48 hours of symptoms to have an effect on patients. So there's this very short window in flu. And so to get drugs to be effective, you have to treat early, and that means you've got to be safe. So I think remdesivir will be replaced over time with safer, more potent drugs.
Okay, if there are no further questions from the audience, I will ask one more question. So to what extent the panel feels that coronavirus is much like flu? Maybe we have to vaccinate every year, maybe we have to treat them slightly bit differently, or do you think if we develop drugs today for coronavirus of today, are we safe for the next 10, 15 years?
Wow. It's coronavirus. I'll have a first go, but just one aspect. Coronavirus is not like flu in the fact that flu has a segmented genome that can mutate and shift and drift, as the old expression is. And coronavirus doesn't do that. In fact, it has a repair enzyme that keeps the RNA stable. But, you know, the data I always quote to people is only half the people who should be taking the flu vaccine actually take it. And of the people who do receive it, it only works on half those patients. So we've got a way to go in flu. I personally don't think we'll see major shifts and drifts like we do with flu, but I think SARS-CoV-2 is going to be with us for a long time, irrespective of whatever treatments we come up with.
Great. And along the same lines, the treatments that we are developing today will be useful to have a less of a patient burden going forward. That is the optimistic message that I collect from this panel discussion. You all agree with that?
Absolutely.
Yep.
Great. So, okay, my clock tells me that I have 3 more minutes to finish. Max, is there any burning question from the audience that I cannot see on my blip here?
Not from the audience, but I'm interested in what people have made from the IL-6 antibodies, and this is obviously on the immunomodulation. We've had a lot of data that suggests that if you know, initially the Chinese obviously thought that Actemra from Roche would be a very successful product in this area, and they were using it, I guess, off-label. And then we've had data that some studies show it does work, and some studies show it in different settings it can work. Is there a role for IL-6s in COVID, or is it just the data is not convincing enough? I'll go Gilead. Why don't I'll ask you if you've got a view on that?
I think it's currently been shown that as a standalone approach, it's probably not sufficient to prevent the progression of the disease. It still may be an important component in the overall treatment of the patients, together with antiviral activity. And indeed, we believe that we do have normalization reduction of IL-6, TNF-alpha, and other cytokines that could benefit patients further on top of the viral activity and inhibition of viral activity that the drug has. So certainly it can be combined, but probably not as a standalone.
Okay, great. On that note, I want to thank you all for your excellent participation and input in this matter. I briefly want to summarize on an optimistic note as I hear from this meeting. So I understand there is extensive collaboration, and we have put in a lot of speed and money into therapeutic development here. And there are many small molecules are coming, but people are not only thinking about the current COVID-19, but they're also thinking about the pan-COVID. This is coronavirus. This is an important development, so that we will be prepared for the next pandemic, hopefully that will never arrive. On that note, so I'm hoping that in the next 6 months, we will turn a page and really we all go back to work and begin our socialization with each other in a much more pleasant way than having to meet in the Zoom all the time. And thank you all once again for your participation.
Thank you.
Thank you.
Yeah.
Bye, everyone.
Yeah.